From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 84, recorded on November 8th, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Good to be with you back live on the YouTube. This is feeling good.
The YouTube.
Flying by the seat of our pants.
Yeah.
Greetings, everyone, from LD Lab Studios here in Salt Lake City.
Looks good there. Looking good.
Yeah, we're doing well. Got a little early winter storm coming in next couple days. I'm hosting a guest from NYU, kind of from your neck of the woods or nearby. Victor Torres is coming to give a seminar for our microbial pathogenesis symposium. And so,
Great.
He's a New Yorker, so he could put up with some snow, but might be a little kind of wintry surprise there.
Look what I got here, Nels. Look at this. See this?
Oh, wow. Yeah. Cast member
caps. I'm going to send this out to all the hosts of Microbe TV. You know, that
beautiful.
I took that from the Disney, right?
I love it. Yeah.
Cast members. So I don't think they're going to sue me. So
I think you're safe, but I'm going to keep an eye on my mailbox. I hope I'm considered.
Would you wear it?
Yeah, absolutely. Count me in.
And I'm hoping to have people one day helping me at events, and they would wear this and T-shirts. I got some T-shirts here. I made two with the same thing, so it's fun. It's always fun to do shit like that. All right.
Good upgrade. Yeah. So Vincent, how about you? You've been on the road some back in New York. What's the latest?
Oh man, I was on the road for 3 weeks.
Wow.
Holy cow! I'm really glad to be back. When did I get back? I got back on I think Thursday.
Gotcha.
Right? And then Friday I went to the incubator and did a TWiV, and now this week. The rest of the year, I think pretty much maybe some travel. I'm not sure, but it'll be good to stay here.
I hear you. Yeah, I've been kind of feeling like this fall was sort of the relaunch of the academic season where we're back in person. Obviously, the pandemic's still going to some degree, but wow, big transition and sort of actually, I mean, even the small things that you almost take for granted. So for example, most of our meetings have been on Zoom up until now.
Yeah, sure.
And so I look at my calendar and, usually it takes me 30 seconds to get to a Zoom meeting, but it might take me 10 minutes to get it halfway across campus to an in-person meeting. And so I feel like I'm always kind of one step behind as we.
Well, I taught in the spring semester in person, so I'm kind of used to it. And I've been going to some seminars in person and so forth. So I'm used to it already. Next semester I look forward to teaching in person again. Should be
very cool. I hit the road a little bit. I gave a seminar at Penn out in Philly a few weeks back, and then I think we actually were probably briefly in the same place at the same time, Washington D.C. I was there for a study section. First one in person. I think NIH is doing a third of them right now in the fall review season in person. And, I think if there's no super spreader events in the headlines, then they're going to go offline or back to Zoom, I should say, out from person in the winter, but then maybe returning in spring. But I do have to say, like, being back with colleagues, with scientists in person has been just a real, really
it's good.
Inspiring. It's really good. Yeah, there's a reason we do this.
Yeah, I've been to a bunch of big meetings with ASV. I went to ID Week. I went to ASTMH. And you can see people are very happy to start interacting.
See people,
with each other.
3D.
And people are talking more than they ever did before. So it's a lot of fun. I enjoy it.
I agree. Yep.
So let's see who we have here. We have a small crew here today, but that's okay.
Fantastic.
You and I like to talk no matter who's listening. It looks like we have Franz. We have Franz here. Welcome back.
Welcome.
We have Barb Mack from the UK.
Good to see you.
We have Philip from Wales. Very good.
Welcome.
Simon is from San Jose. Okay.
Raining. Yeah, we're getting some of that rain. The
Yeah.
atmospheric river coming in, and yeah, Thanksgiving opening, early ski season, both on the Mountain West and West Coast. Yeah, pretty good.
Mm.
We'll take it.
And we have MK from Eastern Massachusetts.
Hi, MK. Welcome. Raking leaves out there. Hasn't it been warm on the East Coast in the last few days, or am I?
Yeah, it's been in the 70s. And you know what they
Ooh.
call it now? This is pretty funny. The Paxlovid summer.
The Paxlovid summer.
You get it, right?
That's right. The rebound.
The rebound summer. We used to call it Indian summer, and that became politically incorrect.
Yeah.
So now it's Paxlovid summer, which I think is just brilliant. I love
like
when
that.
people are clever, don't you?
Yeah, that's a good one.
Rob here. How different does everyone seem in person since you last saw them?
Oh, yeah, good question, Rob. I would say, I don't know if I speak for the others, but I can tell you how I'm different. I've got a lot more gray hair in my beard. I've kind of, added a pandemic on some pandemic pounds. I feel like I'm kind of just still coming out of hibernation a little bit over here. But I have to say, like, yeah, being with friends and colleagues, like, that's just a real boost to the practice of science. And, we have now a whole kind of cohort or generation almost of grad students and other trainees in science who, if you started grad school 3 years ago, let's say,
yeah.
what you think science is pretty different than
Sure.
a lot of us old timers. And so it's a pretty eye-opening experience, I think, to get in person and actually socialize a little bit as scientists.
That's true for those people who haven't been in the field long. But even I am very happy to see people in person. And I went at STMH. We had a booth. Daniel Griffin rented a booth on the exhibit floor, and
Cool.
I'm just sitting there, and dozens of people are coming up, thanking me and wanting pictures. And I think it's great to see people again. Right?
Absolutely right.
Let's see. Lots of Brits today. That's from London. Yeah.
Wet out that way. That's good news, actually. I don't want to give away our paper yet, but there's going to be a definite London connection that we'll see here.
Cool.
We'll rewind a couple hundred years and see what's going on.
Terry is from Virology at Charité in Berlin, which is where Christian Drosten is. Welcome.
Yeah, the Max Planck. Welcome, Terry.
Cherry is from the north of England.
Fantastic.
And MK says the Greeks call it donkey summer. Well, I actually think Paxlovid summer is going to stick. I really like that.
Certainly for the virologists, I would say so.
Elizabeth is from West Virginia. I'm working, won't be in the chat much. It's okay. You can always play it back.
Absolutely.
Later.
Thanks
Elizabeth,
for being here. Yeah, thanks for being here.
Appreciate you. Pete says, my local archaeology society is still only doing Zoom talks.
Yep.
Yeah, but the thing is, it's easy to do a Zoom talk. So I'm doing one at 6 p.m. to an ASM chapter in Pennsylvania, right?
Cool. Yeah.
And I don't have to go there. But more and more, I'm booking like crazy for next year. So, the booking requests are coming in strong, and I'm very happy to be able to do that. So,
that's great. And it's a mixed bag, or there's trade-offs here. So when I was doing a seminar a couple of weeks ago, it was a hybrid, and the advantage was there's more people in the conversation. It's recorded. It can have a longer reach, and I like that. The downside was, like, the way that AV audio was hooked up was kind of a mess. There was a massive delay on my slides. I had to kind of work through that, and sort of both the Zoom and in person. It sort of adds a degree of difficulty, but I do think some of those trade-offs are worth it if we can extend the reach of the science to audiences that might not otherwise have been included, sort of in the before times, before the SARS-2 pandemic made us all Zoom aficionados here. So, yeah.
So our first moderator is here, Les. Welcome. Thank you so much for coming.
Welcome. Good to see you, Les.
Yes. Our moderators are thankless because they do, the job that no one wants to do, but we
super important.
Yes. Appreciate it. Here Agreed. Is Nafiu from Nigeria. Very
welcome. Good. Zamfara State. Fantastic.
Ever been to Nigeria, Nels?
No, I haven't, but sort of embarrassing. I've never been to Africa. It's on my list of I would
Me
love to
too.
get out that.
So
Yeah, yeah.
Daniel's going to Uganda next month,
Yeah, there's a big
and
meeting out there. So one of the postdocs in my group is also slated to go
well, I'm
to
going to
Uganda.
have him take me on some of his travels in the future. Going to bring some recording equipment and do some local pods. I think that could be very interesting. Yeah.
Great idea.
Right.
Yeah, yeah. Welcome.
Well, anyway, welcome everyone. Thanks for coming, and we're gonna talk about an interesting paper that Nels picked up today, which is just it's in the line of what we do here on
Eh,
TWiEVO, right?
Yeah, absolutely. I was a little cautious here. So when we pick papers, in this case, it sort of fits my sort of professional interest here. I don't want to oversubscribe us in that territory, but I think this one is really worth taking a deeper dive on. It's for reasons that hopefully we'll get into as our conversation unfolds here. So the paper is entitled Evolution of Immune Genes is Associated with the Black Death. This came out a couple weeks ago, I think mid-October, end of October. And, well, before we get into the sort of concepts and the ideas here, let me just give a introduction to the authors. So some shared first authorship between Jennifer Klunk and Taurus Vigalis, groups both from Canada. This is the McMaster Ancient DNA Center in the Department of Anthropology, Biology, and Biochemistry in Hamilton, Ontario. And then sort of complementary team at the University of Chicago. So Jennifer from McMaster, Taurus from the University of Chicago, and the co-senior authors are Hendrik Poyner, who's at the McMaster. I think he runs the Ancient DNA Center there. And Luis Barrero, who was in Canada, had his lab up in Montreal for a number of years, might still have a little bit of a remnant there, but has moved his operation to the south side of Chicago at the University of Chicago. Kind of an impressive set of affiliations here. They're into the high 20s of footnotes, which reflects some of the many affiliations. Yeah, that's like half the page is where these folks are from or the places they associate with. So the Barrero lab at the Department of Human Genetics, but also all these committees at Chicago, and then sort of a real team effort here. And I think what we'll see is some really cool interdisciplinary, multidisciplinary approaches to linking the evolution of some immune genes, specifically to one of the outbreaks, one of the bigger pandemic events of the Black Plague, the bubonic plague caused by Yersinia pestis, a sort of notorious bacteria. So we've talked a little bit about some work from Luis Barrero's lab.
Pandemics, man.
Oh, geez, yeah.
These things kill almost half the population.
Correct. And so now we're talking about, when we think, so yeah, I think that's exactly the right thing to bring up at the top is like to, put some calibration here. For example, with our SARS-2 pandemic, sort of trend lines willing, an absolutely terrible last few years, but nothing like 30 to 50% of the population being killed with some of the more pathogenic strains of viruses, or in this case, of bacteria. And so, we'll get into some of the details of this. I think even you had highlighted there, Vincent, in the abstract, that first paragraph where it says. I wanted
to ask you.
Yep. Yeah.
They say infectious diseases are among the strongest selective pressures driving human evolution. I mean, that's a TWiEVO statement, right?
That's a TWiEVO statement.
And so I wanted to ask you, is that true, and how do we know?
Yeah. So I think it is. And they cite a couple of papers. Worth it to pick this one up, by the way, for our audience, if you have interest in this area, so we've made, and I think, a lot of the work in the field from my lab as well, thinking about these, kind of molecular arms races or ongoing genetic conflicts. We kind of put up some of these ideas, but we're really looking kind of from a phylogenetic perspective that might kind of go over millions of years in a lot of these cases, where we'll sample some modern species today, including our own humans in some cases, but then we're really working backwards to try to infer, what are the sort of genes that are evolving in ways that sort of reflect these kind of cat and mouse games of adaptation And so, um, and so some of that work is sort of, it seems obvious that this would, since we're part of all of mammalian evolution, that this would apply to humans as well. But I think what this paper does really in a semi-convincing way. There's a few. We'll do a little podcast peer review like we always do, and bring up some some of the strengths and weaknesses which any study has. But to really kind of try to pinpoint a genetic event, and so that depends on exactly, that what you were mentioning, Vincent, the fact that half the population, up to half the population was being killed, or the mortality rates that were. And so at those kind of levels, you could imagine that it could influence what are the genetic backgrounds that make it through, or which ones don't. And so that's kind of where that idea comes from. But there haven't been a lot of like direct cases where you can kind of try to pinpoint the genetic changes to those events. And so the Barrero lab has been doing this for a while, and kind of looking at those deeper evolutionary timescales. We talked about this, Vincent, in TWiEVO number 8. Everyone's a little bit Neanderthal. And this is obviously, not our species, but some forerunners or cousins from way back in the day, and how there's introgression. Some of the genetic variations, in this case, an immune system gene called OAS, might have come over, and that could influence actually immune responses today. But these are events in the deep past, and sort of this mysterious stew of infectious microbes, viruses, bacteria, fungal pathogens, parasites that are somehow influencing all of this. And so, what we would really like is to start to, or in addition to this, it still is a very fruitful and interesting area of research. A little, let me also identify my conflicts of interest, since that's what I do for a living. But to really, and especially in human cases, to sort of put that in a sharper perspective, or to take a more pinpointed view of this. And so that's what this gets at. So they've decided to take a look, or see if they could do that, using our more recent history with the bubonic plague, the Black Death. And so this is the first outbreak of the second pandemic. So the idea was, I think it was in 500 AD, that there's thought to have been based on sort of, historical or archaeologic evidence. It's thought to be the first bubonic plague. Then things get silent for like 800 years until the 1350s, and that's the time frame that we're dealing with here, and it's happening in Europe. So to all of our listeners here in the UK, basically on the ground you're standing on around London, they actually were able to get ancient DNA samples from some of the cemeteries, and related even mass burial sites that have been linked to the Black Death, as it was called then, and I think the authors made some really clever choices, or logical or reasonable choices, in terms of thinking about, okay, can we actually, is there enough information here? This is recent enough in our history that we can identify samples, and by samples, they're taking bones from skeletons, and you can see that in Figure 1 of the paper. There's like literally a photo of the mass grave site, and so they're going through. There it is up in the upper left corner, as Vincent's pointing out there. So they're taking some of the long bones, some of the other bone samples here, and seeing if they can recover viable DNA from them. And so we've talked a little bit on some TWiEVO episodes about doing this to actually pull out sequences from the microbes, things like smallpox, variola virus that causes smallpox. In this case, though, they're really interested in the immune genes, the immunology here. And are there genetic changes that could underlie whether you are more likely to survive or succumb to the Black Plague? And so that was really, I think, in the case of the system they chose, they had this really great access to these samples where people had died either before, during, or after the Black Plague. And so being able to identify that actually allowed them to do some population genetics, which sort of is the main strength, I would say, of the genes they proposed to be involved. The genetic changes that they proposed might have been involved in whether you were susceptible or resistant to these infections.
You also had some samples from Denmark to kind of
exactly
confirm, right?
Yeah. And so I think they make the point that the amount of sampling and sort of the archaeological framework in the samples from London were strong enough that they used that as their first one, but then do a really clever, so they got a couple, we'll get into the nitty gritty of what they did here in a moment, but then using sort of the idea that if this happened in one location, it would probably like this, if there, you see the same genetic differentiation happening in another population, just as you're pointing out in Denmark, that could really sort of allow you to zero in on high priority candidates. And so they go from something like 200 candidate regions in the genome that they think might have been under this selective pressure because of the Black Plague, to 4 that overlap with the group or the samples that they analyzed in Denmark.
So Priya has a question here. She wants to know, is this what Richard Dawkins writes in The Selfish Gene? I think she's referring to this idea of positive selection.
Yeah, so that's, it's certainly related. There's another Dawkins book that I'm not going to be able to place the title that might even get closer to some of these ideas. But yeah, so if we take like a gene's eye view here, what you can imagine is that, so, we're carrying around a collection of whatever, 23,000, 25,000 genes. And if one of them encodes a variant that makes it more likely to survive, that's from a gene's eye view or from a selfish gene's view, that's an advantage. And so, what we're kind of selecting towards is persistence or existence, or even, to dominate a population. And so you can kind of think about it 2 sides of the same coin. One is that sort of on the positive side, if you encode a change to a gene that means you have a better immune defense, that will be selected for. Whereas if you have a variant of a gene that actually makes you vulnerable or sort of contributes to some vulnerability, those are the folks that might be lost from a population, from a host population, and then that gene will become less frequent. So if you think about it from a gene by gene basis, these are all tied together as part of our genomes or in physically linked in chromosomes. So things get a little bit more complicated in a hurry in that sense. But yeah, you can kind of, I think that's a good, it's worth it to kind of think about it at that level. That's simplifying in some ways there, but that makes some sense. Yeah.
So Steen points out that on TWiEVO a year ago, we discussed some ancient DNA work on Viking Virology.
That's right. Smallpox of the Vikings. Yeah. And I might return to that. Thank you for bringing that up, Steen. I'll probably return to that at the end here, because I'm actually, and maybe there's some more work along these lines. I think this study actually proposes a nice roadmap where you can think not just about the microbes or the viruses genomes or sequences that recover in that case, but if you can do that sort of dating in that sort of fine scale, you might even be able to pull out, was there a sweep happening with smallpox? And that's another one, just as you're saying, Vincent, so 30 to 50% mortality rates was not unusual for smallpox as well. Sure. So another consequential one. I wouldn't be surprised if there's something, I don't have any knowledge of it, but something percolating on that front as well. Yeah.
Now, Terry mentioned CCR5 delta 32. This is very interesting because this is a deletion in the gene. It's been fixed, but we don't know what the selection was that fixed it.
Well, but there's been some suggestions, and I'm probably not going to do justice to it just off the top of my head, but that actually the Black Plague was involved in that. And then this gets,
even
smallpox.
I mean, basically all the big ones.
That's right. So that's, but that's this illustrates that, does the evidence really match the claims in some of these cases? And I think let's see what this study does in terms of maybe a different gene that will focus on not CCR5, we'll focus on one called ERAP2, and we'll get into the details of that. But it raises a second really good point here, which is that, the trade-offs that exist or the sort of knock-on effects that can happen. So you might select sort of an ancient population on one allele. So that delta 32, that sort of, technical jargon is a deletion of a little chunk of that receptor that then in today's world of infectious microbes means that it's harder for HIV to enter cells that have that deletion. And in fact, the first functional cure of HIV was this accident where a cancer patient got a bone marrow transplant with a donor who happened to have that deletion. And then even though they were infected with HIV, they were able to clear the infection, we think, completely. This is the Berlin patient. So we're already kind of in the mix here. We're in the heat of the battle with like, what do these sort of mutations mean for genes moving forward?
Isn't it interesting that, that delta 32 could not have been selected by HIV because it's ancient. It's been around for many thousands of years, right? 4 to 16% of humans have it. So there's no way it happened in 1920.
Nope.
But it's really interesting, whatever selected it, that it also affects a different pathogen, right? Which we'll see today as well.
Correct. And this is like I would say that one of the big themes, one of the big concepts in the field, or the reason the notion of a arms race works, is that the infectious microbes sort of show up at the same interfaces again and again. And then our evolutionary analysis is able to tease that out. So we're definitely better at getting to these like kind of repeated events than we are to the one-time events. So, okay, but let's dig in on this one. So 206 ancient DNA samples from before, during, and after the outbreak of the bubonic plague around 1350 in London and in Denmark. Those are where their samples are from. And so the first thing they do is they kind of take it's hard to get good genetic data from samples that have been around for hundreds of years. And so, they take a little bit of a compromise approach, and I think a very reasonable one, which is to prioritize on immune genes. But I think this is worth keeping in mind, just as sort of the big picture of this study, is that what you would really love is an unbiased whole genome approach. And if you see really strong signals in genes that you aren't picking, there could be some real action there, that you have a little bit of a blind spot. But in any study, you have to make compromises. So they pick, I think, something like 300 or 350 different gene locations, immune genes. And what they're looking for is whether there's evidence that the gene there are different alleles. There's differentiation. So you don't just have sort of the random variation here, but you have what looks like parts of sweeps. So an allele, if we go back to CCR5, that delta 32 was at 5% in the population. If you come back in 50 years, is it now at 80%? That would be an example of that allele starting to potentially sweep, or patterns like this. So the real trick is that, mutations are happening constantly. And so how do you identify what looks like a real sort of selective event where either, I'll probably use interchangeably, which is not quite right, but the idea of a genetic variant or an allele, we're talking about, in this case, is really SNPs, single nucleotide polymorphisms. These are single letter changes in that 4-letter DNA code. A, T, C's, and G's are getting mutated or changed with some, and most of the time there's no consequence to that. Mutations happen. But what the authors really want to try to do is prioritize cases where they think those mutations are actually being selected on, and there could be an immune consequence from this, specifically tied to the Black Plague, given the sampling they've done in the archaeologic studies and the DNA they've recovered. And so sort of important control here, Vincent, is they pick about, I think, another several hundred regions that they sort of designate as neutral. And I didn't go quite into the, how you do that is important, because that's sort of your comparison set. You'd say that sort of represents kind of the noise of the mutational events happening. And you want to then use that to benchmark across your candidate genes. And so they go ahead and do that using kind of these PCR probes. So this is amplifying short regions of DNA from these ancient genomes, doing a bunch of sequencing, and they get roughly 5x coverage for each of the several hundred regions of the genome that they're trying to genotype, or to try to define what the mutations are. And then we already mentioned, so I think they pull out a couple hundred that they think are, could have something going on there, meaning that before, during, and after, the frequency of that allele or that variant changes. Either it goes on the upswing, which might indicate that there's some protection conferred by this thing, or it goes on the downswing, meaning if you have this, you're vulnerable. In some cases, that's a different allele of the same gene. And so that would, be consistent with that hypothesis. 250 candidates is a lot, though. And so this is where they sort of, go to that idea that, well, if this happened in London, it was probably happening in Denmark, same time, very similar genetic background in the populations there. And that actually allows them to narrow down pretty quickly to, I think, 4 candidates. And then the one they focus on is a gene called ERAP2, ERAP2. And actually, before we kind of zero in on that one, they mentioned that actually out of all of their hundreds of candidates, a couple hundred candidates, none of them is a mutation in a coding region of the gene.
It's so interesting, right?
Yeah. And so, in kind of the parlance of host pathogen arms races, often it's these, we find coding differences, because if you change the shape of a protein, that could be really pivotal in whether your offensive weapon, if you'll forgive the sort of loaded term here, can work effectively, can recognize and work effectively against a virus, against a bacteria, or vice versa. If there's some pathogenic recognition or the infectious microbe is recognizing the host, it's often through these protein shapes and these interfaces. Of course, the spike protein of SARS-2 interacting with the ACE2 receptor, those kind of non-synonymous or coding changes we see constantly. They don't see anything. So this is puts you in an interesting category of, well, there's two possibilities here. So one is these are all regulatory changes. They involve differences in the expression up or down of the genes. But it also highlights another important thing here that I think comes out later in the paper, which is all of these sort of changes that they focus on might be, so when you're doing kind of these association studies or linking it to regions of the genome, you might not actually have the causal variant. It might be a hitchhiker. It might be a genetic variant nearby. There might be a more complex genetic sort of basis for these changes happening. I think that's the case, including for ERAP2, which they sort of airbrush over a tiny bit. And we'll get to that. But so in the case of ERAP2, so this is a protein. We should talk about what it does. So this is a peptidase. And
in fact, there's no SNP in that. It's just an associated non-coding change, right? Which is like RS2549794 or something. It's close to it, right?
Exactly. Yeah. And so they do this, and I'm probably skipping over here. Yeah. Actually, why don't let's do the genetics, and then we'll get to the sort of molecular biology or the function a little bit later. So they do, so first, why is it that they care about this one, the SNP? So this like alphabet soup of a single genetic change. It's because it really sings out in their population genetics analysis. So they're doing FST measurements or quantifying this sort of metric of differentiation. And actually, if you look at I think it's figure 2. Can we pull that up? So there's some good graphical representations here. So if we look we'll start with C. A and B kind of look at population, like their whole set of data, and it's okay. But I think C starts to like focus in here. So you see on the x-axis here, going from left to right, these are the chromosomes. So this is the entire human genome here. And then on the y-axis going vertically is this FST measurement. It gets complicated in the population genetics, but it's a sign that gene in the population is differentiated from others. So like next to all of those quote unquote neutral areas. And so the orange dots here, these are the 4 that came through both in their London sample and their Denmark sample. And you can see the highest FST is for ERAP1. ERAP2 is another complication. There's more than one of these things in humans. Turns out there's one in mouse. There's been a duplication event in our ancestors since our last common ancestors diverged from the rodents. And so that's sort of the energy here. There's another way of looking at it. Like this is worth spending a second. If you're looking on your screens here, is in figure 2D. We're now looking at, so there's the blue are samples taken right after, whereas the red are samples taken during. So on the x-axis from left to right, it's before, during, and after. And in blue and red are the 2 alleles or the variants. So you mentioned that the RS5 number. This is one of these changes here. And so you can see that from before to after in blue, there's this sort of massive uptick in allele frequency. It went from about 40% of the population had this mutation in ERAP. And then by the end, 70% had that. Whereas this other allele sort of bounces around, and actually even during the Black Death, during the bubonic plague, goes down a little bit from 40% down. So it's that differentiation that they're really looking at here, and that sort of really got their attention. You can see we won't linger on this, but for their other 4 candidates, you also see some patterns. So like the red's going down and the middle 2, the blue's going up in some cases, but down. It's complicated. And so actually teasing out, I think maybe one of the take homes for me here is that genetic data is noisy. We have, 3 billion base pairs. And so you're really trying to tease out you're trying to discern signal from noise and tease out these sometimes subtle differences. But when you go from 40% frequency to 70% frequency in a population over that short of amount of time, I can't remember. Do you remember actually offhand, Vincent, the amount of time sort of inferring here. That's a pretty wild change of allele frequency for the human population. We generally think of ourselves as sort of slow evolving from generation to generation, but that standing variation is being sort of removed in sort of one fell swoop over this huge mortality event. So they calculate what's called a selective coefficient here based on their data that they quantify, and it's they claim that it's that coefficient, it's the strength of selection is higher than anything that's been documented so far on the human genome. And so this gets back to that first point about infectious microbes causing some of the strongest selective pressure on hosts. There's an example of it. We've certainly, I think, in the SARS-2 pandemic, seen the flip side of the coin on the virus side, where these populations can rise and fall in months, sort of right before our eyes, given the high mutation rates, all of the selective pressure of the immune system acting on the microbial genomes or the viral genomes.
So the time now is the pre-Black Death is around 1000 to 1250 AD.
Yep.
And then this is London, and then the post is 1300 to 1500-ish. So that's, span.
Yeah.
What is it? 1500 years, 500 years, basically.
Yeah. And for human evolution, that's right. But kind of a blink of an eye for human evolution, a few generations. Okay, so that was the evidence that they used to sort of narrow down and then kind of nominate ERAP2 as an important outcome here. So I've already been talking a little bit or hinting at that these aren't coding changes. So what's going on here in terms of kind of the going from genotype, a single point mutation that's not changing a coding, an amino acid identity or the shape of the protein, to the sort of phenotype? What's the outcome of that genetic change? And so in this case, again, it's a little bit airbrushed. So there's I think that allele or that mutation is associated with another one nearby that then causes a difference in splicing. So our genes are not so simply organized; they're interrupted. It's kind of a mess. It's like your fragmented hard drive that you have to splice out the introns to get your actual data, which is the coding region of your gene, together for translation. So you go from the DNA, we're going from the DNA to the RNA to the protein. And in doing that, there's this sort of complex machinery that's going in and clipping out some of the RNAs or splicing together the exons from the introns. And so the mutation that they're focused in on ERAP2 is actually interfering with that splicing process in a way that you end up with the exons kind of messed up or put together so that you end up with a truncated protein, or even before then you end up with a sort of aberrant message. And we have all these quality control systems in place. You don't want to just launch sort of junky proteins into the cytoplasm of your cell. Things can go off the rails pretty quickly. And so nonsense mediated decay is a way that you actually can, if these aberrant messages are being made, we can go in with some enzymes that digest away that RNA. You don't even make that protein in the first place. And so the outcome of this splicing mutation is that you turn down the volume on the expression of ERAP2, the protein would go away. And so I'd say in the parlance of genetics, we would call that a hypomorphic mutation. There's less of this protein, the gene encoding this protein to actually act or to do its job. And so now if that's true, then how does that link up now to an immune response or a difference in immune response to the plague? And so now I think is probably the right time for us to talk about what ERAP2 actually does. And it's an aminopeptidase. Do you want to run us through enzymology 101, Vincent, on what amino?
So aminopeptidase is an enzyme that cuts proteins from the amino end, right? The N-terminal end, rather than from the C-terminal end. So cuts it. And in this case, these aminopeptidases, there's ERAP1 and 2, makes peptides that are presented on MHC molecules, major histocompatibility molecules, to CD8 cells. It's part of the recognition that a cell may be infected. So these aminopeptidases are important for an immune response, basically. And so they say, these proteins have major roles in antigen presentation. So we find polymorphisms near the encoding genes that are associated with a variety of infectious agents, not just Black Plague, as we saw in this paper as well. So,
Yeah, that's right. Yep, and that's an important point. So, they're really trying to use the archaeology here to pinpoint those events, and I think they do a nice job of it. But it's important to keep in mind that that's not the only microbe in town. Yersinia was making a huge impact during the outbreaks of the bubonic plague, and it's still around actually, not at the pandemic levels like this, but um, there were plenty of viruses, other pathogenic microbes in the mix as well, and so that's going to be part of the what's complicated about the signals, or interpreting them from the genetics all the way through to the immunology, is that there's a lot happening, and so it's good to keep in mind. And also, we've now sort of honed in on one genetic change, which is wild, right? One, or a handful of mutations, a variation here in this one gene. But also, I think important to remember that our immune systems are encoded by hundreds, even thousands of genes, sort of in these, and we're facing these microbes with this whole array of defenses and counter defenses and everything else. And so, always kind of keeping that back of the mind. But yeah, so the fact that you're potentially changing how you're processing the peptides. So the idea is that the bacteria, as the immune system is perking up and trying to get into the game with it, part of that process involves these aminopeptidases that take some of the bacterial proteins, cleave them, just as you're describing, Vincent, or kind of chiseling away at them so that they're presented properly to this other protein machine, the MHC, which sits on some of our immune cells, that allows this recognition of the T cells to kick off to say, hey, if you see this shape around, we're mounting an adaptive immune response to anything that's like that. We're marking as an invader here that we want to eliminate with sort of the multiple prongs of attack of the adaptive immune system.
Yeah, it's they do an experiment which is not really that satisfying. They deplete mice of CD8 T cells, and they die a week later after Yersinia infection. But yeah, they would die of most things. Right.
I agree with you. Yeah. So I think for me, the real advance here was sort of linking the history and the archaeology to the genetics, and then they kind of do sort of let's get it over the finish line experiments in a sense. But the bigger question is, are they onto something? And is this a useful concept for doing some more studies like this as well? And they don't stop actually with just the sort of direct implication or the idea or hypothesis that the aminopeptidase, like that it's all CD8 presentation. They kind of take a couple other approaches, slightly different experiments, and they note that if you just measure some of the kind of immune responses, it's different. But whether you have the quote unquote protective allele versus the vulnerable allele in ERAP2. And so they look at cytokine production. This is in figure 4. And it's not a simple outcome. So, you might think, oh, well, if you're vulnerable and you've got that genetic background or that allele. And by the way, they're able to look at, here we go. So we've got, so these are samples from people with ERAP that's either one variant, the TT, which is thought to be vulnerable, or the CC in ERAP that is thought to be protective. And then the heterozygotes, people who have one of each. And so you can kind of see these, you kind of step down actually in some cases. So there's actually less of this immune signaling molecule. They look at, so panels A, B, and C are 3 different ones. And then CCL3, which is another immune signaler, that's actually up. So it's kind of a complicated outcome where with clearly different and consistent with sort of that model of heterozygote being sort of intermediate phenotype. But you're not just turning everything, you're not turning the volume to 11, you're sort of turning the dials in different directions depending on the immune signaling. That bigger one in graph in panel 4E is another experiment that I thought was pretty interesting as well. So this is actually looking at how many, kind of maybe a more direct measure on what's the implication for how the immune cells are acting in the presence of Yersinia pestis, the cause of the bubonic plague, the Black Death. And so they're looking at the percent of bacteria killed on the y-axis, again for those 3 sets of alleles, and noting that if you've got the protective, you kill more of the bacteria, whereas your macrophages kill less. And so that's, so that data in particular, I think lines up maybe a little more directly with the impact of the ERAP mutation.
Yep.
Of the variation there in terms of like directly linking that to how well your macrophages are doing dealing with these bacteria. So I think, a set of, in the end, maybe I would grade it a B plus, A minus for the linking it to an experimental outcome. Certainly the experiments they did are really consistent with that being an important part of the conversation for how this goes. But in terms of what that means, for a full blown human infection, that hasn't, I don't think they quite scratched that itch or tried to. That's a really tough experiment. But we've got a late breaking preprint here, Vincent. I just added this late last night as I was trying to brush up on the paper for today's live streaming podcast. There's another group. So this came out, like 3 weeks ago, really recently. And there's another group that just, tweeted. So this is, I don't know if you're familiar with this social media platform called Twitter that has been in the news for for pretty recently. But so this is Gus Hamilton, who is there in Bristol, UK. I think they were already familiar that this paper was sort of, or this work was emerging from the Barrero lab. And so they put up this on medRxiv, put up this really interesting preprint that kind of tries to ask some of these questions about that, again, that ERAP mutation as it relates to, human disease outcomes, actually. Yeah. And so, so they're, so they're looking at, and saying that there's variation has opposing effects on severe respiratory infection and autoimmune disease. So this starts to also get to the potential trade-off here. So, why is it if you've got, better, if you do better, you have a better immune response. If you have this variant of ERAP2 that eliminates the bacteria better or tunes your immune response better, why wouldn't that just sweep to 100% of the population? Why wouldn't we all have that? And so Gus Hamilton's group has been looking at that. You can look at now all of the human population genetic data out there and do sort of GWAS type analysis here. So does having one allele or another link to other disease states? And what's really interesting here is they've got pretty good signal that if you have that protective allele to Yersinia pestis, you're also more likely or more vulnerable to things like Crohn's disease, this autoimmune disease, even possibly type 1 diabetes. And so the idea here is that actually you've kind of, if you have a lot of the ERAP2 peptidase like working away or kind of pulling on the immune responses, that maybe you get an over exuberant response. Everything starts to look like the Black Plague. And this could sort of influence the composition of your microbiome in ways that could then make you more vulnerable today to some of these autoimmune or related issues of sort of balance of your sort of digestive system and things like that as it relates to the microbiome, and who are the constituents that are there if your immune system is sort of overly ambitious to knock these things out. So that's to kind of formalize that, this is this idea of balancing selection, that there's and actually if you kind of zoom up there to the I think you had that figure with the blue and the red on the Twitter page. Yeah. So, after Black Death, the red starts, like, it gets down lower in frequency during the event, but it kind of ticks back up. And that might be a signal of the trade-off here. So, there's actually if you're not under attack in a sense, or you're not in the middle of a pandemic that has a really high mortality rate on your population, then you start to select actually on not having that protection, because it comes at this cost of autoimmunity or an overexuberant immune system. And so that might be what's going on there. And I think a really interesting follow-up study, so what is the value of the results, even if the experiments don't sort of like drive a nail in the coffin of understanding all of the immunology here, sort of, spurring this additional work and allowing us to focus on a really interesting on ERAP2 as a really interesting mediator, the variation there in the human, like, recent past, and then the modern variation that we see today, how that influences or is linked to ongoing diseases
So they
as
do
well.
point out in this Twitter thread that they
Mm-hmm.
find that this sea eel protects against COVID, severe COVID and pneumonia, right? It still has the
Yes.
increased risk of type 1 diabetes and Crohn's, but now
Yeah.
besides being protective against Black Plague, right?
Mm-hmm.
It's COVID, another interesting respiratory infection. So
Yeah.
in fact, the authors of the paper that we're doing did point out the increased susceptibility to Crohn's, right?
Yep. They did.
And
Correct.
it's
Yep.
just interesting that I was going to ask you when I saw that, is there always has to be a trade-off?
Mm,
Is
mm,
it that something that's protective
yeah.
always has a consequence? Is that how biology works?
Ooh, yeah, I love that question. Kind of getting into the philosophy here of that. I would say maybe not like I don't know if it has to, but I think in the vast majority of cases, I wouldn't be surprised because think about it. If just even how much kind of our biology we dedicate like to the immune system, yeah,
Mm,
it's a lot, and so in all of the ways with the kind of defined, kind of lab defined innate immunity, adaptive immunity, but all the way to just, all of the, how our bodies are organized, how our cells are organized. I think if you take that view, all of a sudden, the nuclear envelope becomes a moat or a barrier around our genome. We're trying to protect it, or the all of these compartments of the cell. Are they trying to throw the microbes off the heart of the cell or the gene? Yeah. And so, for me, that the answer to your question is yeah. I think it does. So if you could
yeah,
just take
yeah,
away, if you could take away all infectious microbes, what would happen? I think our immune systems would wither. They would like you would select really rapidly for letting go of this thing that's pretty costly, both in kind of the genomic real estate that you dedicate to it, the energy that goes into transcribing, translating all of those genes into proteins. My God, this is like a
yeah.
lot of our biology
Well, so
there.
the consequence is inflammatory disease, which is what Crohn's is, right? You
Yeah.
have an immune system so complicated, has to be fine-tuned, and if it goes a little off, which is really easy to do, it can be tickled by a different base or a pathogen, then you have
Yeah.
inflammatory disease. And remember now, if you took away all the infections, you'd still have cancers, and we still have to deal with those. So
Oh
I
yeah.
just wonder, cancers tend, not all, but tend to arise later in life, right?
Hmm. Hmm.
So it may be that if you took an immune system away, it wouldn't have any impact on reproductive success,
Mm.
right? Because that happens in the early years of the organism. And really, that's all that matters. Everything else is just us wanting to be 80, 90 years old.
Yeah.
So I don't know that it would matter.
Yeah. Well, I, I think it does. And well, yeah, it doesn't matter in the sense that you'd still be. I agree. You'd still be. Well, if anything, you'd be more susceptible to cancers. I mean, our immune
Yeah, you
system,
would
as
be,
we've
but I
seen
don't
through
think there'd be
immunotherapy,
any
et cetera. But yeah.
selection to fix it because the
Oh, because
organism's
of the
already
age.
reproduced. I mean, I think cancers arise because we live too long, many of them, right?
Many
And
of them, absolutely. Yep.
we don't have them at younger ages for the most part, and we reproduce, and that's it. And we've artificially extended our lifespan, right, by health measures and so forth. So
Oh yeah. Yep.
I'm not sure that there would be a selection to restore some aspects of the immune system. I mean, the point is taken, though, that the infections are an enormous pressure on the immune system to do something. But I just wonder if
Yeah.
it's so complicated that if you tweak it to improve defense, something else is going to break.
Oh yeah, something else will break. I think these are all kind of intertwined, kind of related things. And so in a sense, like the best kind of outcome for us or a host is the immune response is more of a conversation than an attack, right? So it could be, and
Yeah.
so when we think about like the constituents of the microbiome, and I'm kind of off-roading here in a field that I'm not too familiar with the details, but if our immune system just says, hey, is everyone here who's going to just hang out and help us digest food and, be healthy? That's a great outcome. It's when things go out of balance or, and that can happen on both sides, right? So, infectious microbe comes in that's pathogenic and sort of kills off all of the constituents, and then now all of a sudden
Yeah.
the immune becomes less of a conversation and more of a, hey, you shouldn't be here, and those mismatches. And so, I think we're as populations interact and collide, both of the with the same sort of genetic background of the same species and then other ones, it's sort of that constant sort of, there's enough variation that you can get out of balance really easily. And so how much has our immune system evolved to kind of be buffered to that, to have conversations instead of attacks? And how does that balance work out? And so I don't think you're ever, and this is all, I mean, we're kind of talking philosophically, you're never going to get rid of an immune system, but how does it, is there actually selection for it where you can minimize some of these, these massive inflammatory effects? And I think you're right, Vincent. So, part of the conversation here, since we're talking about evolution, has to be what age is this happening, and are you getting through sort of reproductive age? That gets complicated too, because then it's like, how is selection acting on sort of, units of families or things like that, given that the kind of caretaking, as a father of a toddler, the caretaking that our species does with our offspring is,
Sure,
not trivial.
sure.
It's pretty long term, and usually involves people well past their reproductive age.
I
And so,
agree. It's not that simple.
Yeah.
I agree.
But it's fun to talk about. Yeah.
They do point out in the discussion, selection for defense against something like pestis is counterbalanced against costs of immune disorders. And so it's, you can't change one without changing the other. But what I wonder is whether this information, right, ERAP2 is therapeutically useful in any way, right?
Yeah, I think so. So that's, I mean, not directly. It's sort of like, the value of doing fundamental research or doing basic science is that you can really point, so I think what this does really effectively from those hundreds of candidates, it really pulls us one up and says, hey, evolution tells you, pay attention here. This mattered at this time frame. And then, now you can say, okay, well, what is the genetic basis of this? Oh, it's hypomorphic. You either have, more of this protein or less of it. And then what does that mean for, cytokine signaling, for tuning the immune response, or even direct, killing there? So I think what that does is it opens ERAP2 as like an important target to think about, sort of at the interface. And as you're like in this knock-on study, this preprint, that then even goes all the way to SARS-2. This is an important pressure point. Evolution is saying this is an important pressure point for our immune responses, and inflammatory, or immune responses out of balance as well. And so then you can start thinking about targeting in interesting ways and taking more of that kind of almost engineering or more applied approach to think about how do we modulate the activity of this thing to have better health outcomes. And so I think it falls beautifully into that space of basic science, or why I love this, right? So why should we care about human skeletons from 1350? And the reason is because it might teach us something about our immune responses today as it relates to the viruses that we're up against today.
Of course.
That's kind of what gets us up in the morning. I think
just don't know. And I mean, the people who did this preprint on Twitter, they must have been so excited to have their data come up with a similar finding for COVID, right?
Yeah.
Really interesting.
Really interesting. And then they even, go ahead.
I was going to say, Pete Glass said he looked at the map and one of the 3 sites he worked on, but only near the end on early Roman layers. How about that?
Wow. Very cool, Pete. It's a small world out there, from a paper that we pick on TWiEVO to our listener who's chiming. I love that. That's really cool. And even, getting to your point about kind of longevity too. I think in that preprint, they actually make the point that there is, this is from the Gus Hamilton group, that there could even be, some association with how long you live, depending on the allele of ERAP2 you have. Like it's linked. It's not, I'd have to look closer at how good that is, but how strong that association is, but all the way to longevity. And so this is like, in one sense, we've really increased our lifespans, but has evolution acted in ways that reduce it as part of the conversation of benefits and trade-offs? And what a messy system. So, another reason I really like the work, I like the paper, is I think it highlights how, it dispels the idea that evolution is this like beautiful process of perfecting, getting to this sort of like fitness peak where it's all I see it a lot more as chaos of trade-offs, and this got selected on this day, but that will have an echo through human history where that will mean, people might live a lot shorter lives or deal with other issues like Crohn's disease. It's really interesting to kind of how that all kind of swirls together. And evolution is messy, maybe, is one of my take homes from today's conversation.
So Barb Mack checked her DNA sequence. She has the truncated haplotype.
The T's, is that right? So if we look back at, my screens are, speaking of
GG
chaos.
and TT.
So there's, GG and TT, and I just can't remember off the top of my head if that was associated with protection. I think it was. I'm just scrolling over now. Oh no, so I think it's the CC is the protection. So in this case, we would make the prediction, Barb, that you might be a little more vulnerable to the Black Plague, but enjoy, you're better off in the longevity in the issue with autoimmunity. So the good news is, I think if I had to pick a genotype, no question today, I would pick TT with the information I have today, because I'm pretty happy with the massive strides we've made since 1350 in microbiology and antibiotics and things like this, so that a Yersinia pestis infection, I can lean in to a little bit of our medical technology, and not have to depend as much on my sort of genetic background.
Yep.
Also worth it to be a little bit, I love this sort of the DIY genetics as we've done 23andMe, et cetera. Worth it though to, kind of keep the bigger picture here. All of these associations we're making, and we're kind of picking one change out of 3 billion base pairs, and then not to over interpret. I mean, the sort of individual outcomes with the population level, population genetics and the population level outcomes. Like, so when we're talking about the populations and the allele frequencies of 40% to 70%, it's not from 0% to 100%, because all of that kind of population genetics or infectious disease is being litigated in complex humans with all kinds of genes and different backgrounds coming together through different sort of recombination, et cetera. And so even though you might be a little bit vulnerable at that genetic interface with Yersinia pestis, you could have some other variation that actually makes you quite resistant as well genetically, that's sort of not considered in this study. But it's really fun to kind of also just even link it to our own genotypes. But I agree, Barb.
Barb Mack follows up. I do have autoimmunity, but not Crohn's nor type 1 diabetes. Yeah, interesting.
Yeah, there's a lot going on in terms of how our immune system and physiology come together for those outcomes. But that's good to avoid those.
So Rob has an interesting question. Genes don't have emotions, but as a human scientist, how do you feel when you imagine that the history from a gene's eye view?
Oh, yeah. Don't have emotions. And sometimes, as geneticists, this is the trick, right? When we're trying to talk, is we almost become like these. It's hard not to kind of adopt these things, this genetic variation, almost like a friend or a companion, and talk about it that way, to anthropomorphize. And so, so the gene's eye view, I think, is not perfect. So all of our kind of analogies, or the selfish gene isn't perfect, but how do we, talking about or conceptualizing these genetic concepts, or even, just almost kind of trying to come to almost, something like a physics level sort of idea of principles, I think is really difficult. And so I think these all ultimately fail, and you can kind of find exceptions, and the exceptions might be the rule rather than the exception in some of the cases. But the question for me becomes, even if we sort of kind of bend the edges here and aren't formally correct, even in some of the ways we talk about this, is it still useful as a concept to then articulate a hypothesis that we can test in some way to advance science? And it's sort of that sliding scale of truth that we never get there, but are we getting better? Are we getting closer to the biological truth by taking some of these, compromised or hobbled ideas? And for me, in terms of thinking kind of the gene's eye view, and actually there's an entire book by Ovid Argren, who I haven't read the book yet, but we might, this might be a future topic on TWiEVO. And Arvid has actually offered to join us at some point into the future. It would be really, so I'll kind of keep that percolating in the back here. But so he kind of takes up this idea, and this, I think the book came out a year or two ago, and it really takes up this idea, kind of building off of some of the selfish gene from Dawkins and others to see if this really holds up. And I think in some cases it does. But the question then becomes, is this also the way we conceptualize or talk about some of these concepts? Does this also sort of make us vulnerable to blind spots where we kind of miss, because we're kind of putting all of this evolution into this one framework? Are we potentially limiting ourselves and missing all of this other biology? And so that's where I think, Rob, your point here, your question is really well taken. It's always, I think, important to be cautious, open, or maybe not cautious, but to be open-minded, and to let like ideas filter in, or try not to get boxed in or limited by a gene's eye view or a selfish gene, and to think bigger whenever possible.
I'll never forget when I was in college, I learned genetics from Jerry Fink, right? He taught the course. And one day he brought out 2 rabbit pelts, right? One of them was white and one of them was brown. And he said, they're exactly the same genotype. One was raised at a different temperature of the other, and therefore you get a different coat color. And he said, the environment is also important, right? Not just the genotype, but whatever your surroundings are makes a big difference. So you have to remember that. You know, we may have a polymorphism that looks interesting, but there are other things going on as well.
Absolutely. Yeah. And that's, like coat color or these environmental, like, so, turtles, for example, but depending on temperature will dictate their sex. And so under scenarios of climate change, do we end up with populations that crash because they end up being I can't remember the details here, but they're all female or all male, and on a sort of a crash course. But that's evolution again. So as environments change, and there you've got this landscape of mutations, how does selection act to bring some up on different environments versus others, whether that's the climate around us, the temperature, whether that's a set of infectious microbes, and how permanent is that, or how adaptive can you be? That's, a question that we'll be for sure up against, or continue to be up against in some pretty dramatic ways going forward when we think about the environment.
So have you read this Invisible Designer's Brain Evolution Through the Lens of Parasite Manipulation?
Oh, no, I haven't. Yeah. Quarterly Review of Biology. Thank you for the tip on that one. I'll pick that up for sure. I'm doing a little screen capture with my antiquated laptop setup here. Thank you.
Pete has some information on one of these sites, the last World War II bomb site. One corner had a small medieval church, St. Nicholas Shambles, demolished in 1575. One grave had a woman partway through childbirth, right? So I guess the bomb hit and killed everybody. Yeah.
Jeez. Wow. Yeah. Wow. Yeah. Well, that, so demolished, but the church itself demolished in 1575. Yeah. So not my expertise, ancient DNA or the archaeology here, but, this is what's really one of the really exciting, and, we saw actually, even this fall, the Swedish scientists who run the Nobel Prize think that ancient DNA is important too. They gave the Nobel Prize in Physiology and Medicine to Svante Pääbo, who is well known for pioneering work in the area of archaic genetics, of getting bone samples all the way back to the Neanderthals, the Denisovans, and then putting that together. And I think, and so, there's a little bit of interesting controversy. Is this how is this physiology and medicine? But I think this paper kind of helps answer that. We're not going all the way back to the Neanderthals here, but we're seeing genetic differentiation, which has real outcomes for diseases and for physiology in medicine today. And so, and hard fought, I would say, over the last few decades of really getting it right. And actually, so Hendrik Poinier, one of the co-senior author, also well known for his work, pointing out that, hey, it's really easy to sequence a bunch of contaminated DNA from your what you would hope would be your pristine sample. And so some interesting kind of details in the background.
Was the Pääbo Nobel for the technology or the findings, right? Because if it's technology, I would say it should be chemistry, like the sequencing Nobels were chemistry, right? But if it's finding, then that could be physiology or medicine, because now everyone mines the Neanderthal genome and finds cool stuff, right?
Yeah.
But
And I think it's both in this case. It's sort of
Yeah,
a
it probably
hybrid.
is. Yeah.
Yeah. Doesn't have to be either or, but and I don't think, I mean, so the technical measures that you have to take in terms of how you think about the contamination and in
Yeah.
dealing with it,
Yeah.
that's a
Yeah.
pretty that's not a trivial thing to cross
No.
over, and
No.
with a lot of mistakes along the way, by the way. But then I think it's combining it with what you can unlock from that. I have to confess, I don't think I actually read the what the Nobel Committee said in particular to justify the prize, but coming from a TWiEVO perspective, a pretty exciting development, I think, to see that recognition from the Nobel Committee. Yeah.
So they say for his discoveries concerning the genomes of extinct hominins and human evolution. So they say his research gave rise to a new discipline, paleogenomics.
Yeah.
So, I think it's beyond the technology, obviously, right? But what
Correct.
I think is cool is you almost had him on TWiEVO. But he wanted to
we
go
did,
skiing.
yes. That's right. Svante was here visiting. When was this? It was about 5 years ago, I think.
Long time ago,
and I was trying to line him up for an in vivo interview here, and then fell short. Didn't quite happen, but maybe, and I'm guessing actually our prospects of landing him for TWiEVO
is less
probably
now,
exactly. He's probably got an email box flooded with fancy invitations, and so. But yeah, we went skiing about a half hour from Aldi Lab Studios up at the Alta Ski Resort. Had a really fun afternoon. Actually, who joined me on that ski trip was Florian Mattersbacher, who was our guest on TWiEVO last episode, talking about the birds and the special issue of current biology. He actually knows Svante from his own training when he was growing up as a scientist in Germany and interacted with the Pabo Lab many times. And so we all went out skiing for an afternoon. Really fun. And kind of fun to see that.
You've read Pabo's book, right? His autobiography.
Yeah, I've kind of skimmed it. I haven't done a deep dive on it.
I read it. It's an interesting track of. Well, he got interested early on, and he used to get cow livers and stick it in an incubator for weeks to see if he could get DNA out. And he said it stunk like crazy. It's good. It's really a good following. And you notice, you learn how hard it was to do. They had to overcome a lot, and it's very challenging. So, but I think it's not enough to have a good technique, I think
agreed.
Have some results, and they certainly do both. And Peter here says that he stressed the implications of ancient DNA right from the start.
Yeah, I think that's right.
I think that's correct. Yeah.
He's also open. I mean, he confessed to us on a ski lift that afternoon that, the first Neanderthal mitochondrial genome was actually his own, getting at the issue of contamination. And so I think that's another important part of it, is being not being dogmatic about this worked or didn't work. And, so the scientific process, the ups and downs, the fits and starts. And I think that's also really helped to launch that field. And I would say, broadly define this field of paleogenomics. I think the paper we've been discussing today fits in that category. It sort of has that same, what happened hundreds of years ago, not as ancient as some of the ancient DNA work, but can we learn from by taking a genetic view and linking that to these phenotypes, can we learn something about what happened hundreds or even thousands of years ago that influence our outcomes as a species today?
Yeah.
And so I thought it was, so anyway, getting back to the paper, I thought kind of a nice,
very good. I like it.
Yeah, nice one. This is not perfect, but but maybe a nice roadmap for more approaches like this. And I think someone just brought up earlier, this idea of could we look at some of the Vikings samples, the smallpox samples, and see something similar to that? That was kind of my thought too. Are there samples that might where you could use the same framework or some of these techniques to step through and make a case for what did smallpox do to humans in terms of some of our immune, our genetic diversity in that sense.
So then it all becomes a matter of curated collections of ancient DNA, right? That other people have sampled and stored, and you can access.
Yeah, and getting that right, I think that's an entire academic pursuit in itself.
Yeah.
And so to go from that, right, the ability to sample these archaeologic sites, as that Pete who was pointing out, from working there. I mean, I'm sure he could tell us stories about how you don't just show up with a shovel and just start, collecting skeletons. And then once, so you go through all of that professional work to get that right, to get the, to work with folks in terms of the historic preservation, um, uh, dotting the I's and crossing the T's of sampling that in a responsible way, and then getting the DNA out of the samples in a way that preserves the signal there, that you're getting good genetic information, and then taking that and putting it through a pipeline, that all of the analysis, are you doing the population genetics? And so maybe that's the handoff a little bit from the Poinier lab to the Barrero lab is in that part of the study. And then once you've run the kind of FST and related population genetic measures, then thinking about, okay, let's build a hypothesis and test it, even with a sort of an immunological outcome or a microbiological outcome of seeing what this means for macrophages killing Yersinia pestis. And so it's kind of going from that grave site from 1350 to the macrophage. That's I think what's really impressive about this paper.
I like that, and I can't remember. We've done some recent papers that have a similar idea. You look for clues in a genome, and then you come in the lab and do some experiments. It's really good to do that.
Yeah, cool.
So Pete here says, local queen fought the Romans and burnt the nascent London down in 60. I saw evidence of that burning right at the bottom of occupation on GP 75, just north of St. Paul's. Cool.
Yeah, that's really cool. Yep. All right, Vincent, I think that covers it for.
It's good. Thank you, Nels. Very good.
Paper. Oh, it was really fun. Thank you. And why don't we move now to our science picks of the week?
Oh, yes. And I want to just second that the bird TWiEVO that we did was just a lot of fun. And I thought he was terrific. Florian was just terrific. And we had a good conversation. And birds are pretty amazing. And I was somewhere sitting outside and I think recently it was in Hawaii for a meeting, right? And I was sitting outside and a bird sits on a railing. It's just a little sparrow, right? And, it's got that jerky way they turn their head. Not all birds do that, but some have that really mechanical stepped motion, right?
Yeah.
And it twists its head. It sees a crumb on the table in front of me, jumps, eats it, and flies away. It was like 4 feet away from this crumb that I could barely see. So then I started throwing out crumbs, and the birds went nuts coming. And I just loved it. It's so cool. They're so cool.
I agree.
Right?
Yeah,
but then I saw some birds, pigeons with no legs. They just have stumps and they're walking on. I feel so bad for them, right? What the hell?
Urban living. Yeah, exactly. No, and that, we could barely scratch the surface of that issue. Tons of cool articles that I hope folks were pointed to that, have taken a look, or that becomes a great resource. It kind of reminded me. So getting to our picks, that kind of reminded me of, so that issue, I think that was only a few weeks ago. That issue, it's no longer the current issue of Current Biology. Every 2 weeks they churn out a new one. And so the kind of it's amazing just how much scientific information is produced, even in one journal, just over this, that sort of, it's almost to me feels like waves crashing on the shore of stuff coming in. And so that's actually, I have 2 picks of the week, but my first one is going back to Current Biology. So they have in the current issue, not the bird special issue, but the one that's out there now, an interview with Paul Turner. That I thought was really nice. I think Florian runs these interviews as well. So Paul was a guest of ours on TWiEVO 44. The enemy of my enemy is my phage. This is bacteriophage and phage therapy. And so a fun interview there with Paul. And that was, Vincent, that was a great moment for TWiEVO. I think we did 2 in vivo interviews, one with Paul Turner. This was at the ASM, I think, American Society of Microbiology in San Francisco a few years back. And we also talked to my colleague here at Utah, Talia Karasov, who works on the evolution of plant immune defenses, and some really cool work. But so anyway, check out that interview. Check out TWiEVO 44 as well, where Paul sat down with us at San Francisco and talked about his journey through science, some of the work they're doing. Speaking of sort of repurposing some of the diversity around us and then using that in a medical way, I think phage therapy continues to have some interesting, complicated, but interesting potential. Yeah. And then, sorry, I'm kind of filibustering here on picks of the week, but I've got one more, which is, so it's pretty short. This is just, I'm kind of maybe hoping that Twitter will still sort of somehow exist more than a couple weeks from now. It's looking, I don't know, mixed forecast at best, given some of the new ownership that has moved in there. But for me, Twitter in the last, I don't know, 5 years, 10 years has become sort of an easy one-stop shop for interesting scientific tidbits, information, conversations, people putting preprints, kind of talking about the business of science, how we publish our work, think about things like that. So anyway, so I've just got a quick link to a tweet, which is the map of the world if you're a fish. And I think, one of the challenges of Twitter is this, I don't know who to attribute this to. It's sort of the, don't believe everything you read on the internet, quotation attributed to Abraham Lincoln or something like that. But anyway, so it's a map of the world where you just see the ocean kind of in the center, and then all of the land is just at the edges and sort of recedes. And so I kind of like that idea of just sort of switching up your viewpoint a little bit in this fun way. And so we'll put that link up there as well.
It's fun.
I'm done with picks of the week. How about you, Vincent? What's your science pick of the week?
So I've been reading a book by D.A. Henderson, which is the what is it called? Smallpox, the Death of a Disease, right? It's I've had it for a while, and it's very good. Yeah. But he mentions in the beginning that he has a more detailed, free resource. Well, not necessarily him. I think he's one of the co-authors. It's called Smallpox and Its Eradication, and it is available on the WHO website. I'm going to do a screen grab here so you guys can see it. Yeah. So it's a 50 megabyte. It's a big manuscript, but it's a PDF. It's free. It's by Fenner, Frank Fenner, D.A. Henderson, Arita, etc. And it's everything. 31 chapters, definitive history, tons of illustrations.
Wow.
And there it is. It's free. You can just download it, and it's free. And
that's really neat.
Let's see what this thing looks like when it down. It should. The WHO's got slow servers, so.
Oh yeah.
But anyway, I highly recommend it if you need smallpox resources. This is where to go. It's totally free, and definitive history of smallpox from the beginning as far as back as we can go. The end, right?
Huh. Yeah, that's great. Yeah, one of the only ones we've eliminated, right, in our sort of medical history with this massively successful vaccinia virus vaccine. Speaking of, I can't resist, since you brought up smallpox and pox viruses, I guess I'll get there kind of through a circuitous route here, but I wanted to second a really great interview you did recently on TWiEVO with Eddie Holmes. I can't remember the exact episode number here, but I think a really important one for folks who want to so hear from an expert in terms of virus evolution, obviously mostly as it relates to SARS-2. So I really appreciated that conversation that you had, Vincent, that you organized and conducted with Eddie, one of my science heroes. But the reason I kind of was thinking about that is actually there's the poxvirus connection. So now that Eddie's has been in Australia for a while, he was in, speaking of Fenner, was sort of involved in some studies, and I think they might still be ongoing, of a different pox virus that didn't affect humans but affected rabbits, myxoma virus, used as a biological control agent. And so what's cool there, maybe an echo of our paper, is that you've got rabbits from in freezers from, this is like I think the 1950s.
yeah.
From before, during, and after
That's
the
right.
release of myxoma, and then virus samples as well. And so you can play that same game of asking, okay, what do the genetics look like? I think if you were to do a kind of quote unquote natural experiment or biocontrol initiative today, you'd love to have some more evolutionary biologists in the time saying, here's the population we'd like to look at today, how much to sample it to actually get the power to really look at that. But there's a paper or two that gets at some ideas about the immune systems of the rabbits and the genetics behind that, of how that was changing. And so anyway.
Yeah, I think we did a paper on TWiEVO a couple of years ago where some people did that. They took rabbits before, during, and after, and they looked at some, and they found, in their surviving rabbits, amplification of some immune genes, right? So
that's right, exactly.
Suspected. They also have virus samples from before release and then during and after as well. So that's that's good stuff. I really enjoyed Eddie Holmes. We should get him here on TWiEVO and just talk about non-SARS-CoV-2 things, right?
Totally agree. Yeah. Yep.
But we just would have to do it at 5 p.m. Nels, because that's 7 a.m. for
Oh,
him
yeah,
the
of
next
course.
day,
Yeah.
and that would be a good compromise, so he can do that.
Yeah. I'm glad that worked to make that connection. Let's see if we can line something up like that down the road.
So that's my pick, that smallpox PDF. And someone says
cool.
here, you can read Frank Fenner's description of the writing of that book in his autobiography. Okay. Yeah.
Nice.
I have Frank's autobiography. Yeah, I started to read it. I didn't finish it. It's quite good, yeah. We have a listener pick from Larry, from long-term fanboy Larry in San Diego. Boring 70F21C and sunny today, October 26th, but we had our Paxlovid summer last week, almost reaching 100F38C.
Yeah.
Speaking of San Diego, I have to take a brief diversion. You know Matt Doherty, right?
Yeah, of course. We overlapped as postdocs together up in Seattle.
So I was in Seattle last week, and I met one of our listeners who is often here, Lost Laboratory. Right?
Yeah.
And so she brought me to dinner at a little neighborhood outside of Seattle. She said it's a very desirable neighborhood. What is it? Do you know what it's called?
Oh, there's a bunch, but yeah, were you north, south, east, west? Probably not west.
Anyway,
that's the water.
It's where Doherty used to live. Okay.
Oh, yeah, yeah.
And we walked past his old building. He owned a building, right? And she took a picture of me in front of it, because they were friends also. And she sent it to him, and he said, "WTF? What is he doing in front of my?"
I love it.
Well, I'm taking him to dinner. It was a cool restaurant, a hole in the wall you'd never find.
I'm trying to remember. Well, it wasn't pizza, was it? You weren't at Delancey's.
Well,
this
we
might
did
have been
pizza, but I forgot what the name is. She's not here now, but I'll have to ask.
We'll find out. We'll close the loop on that. Yeah. No. So Matt has started his lab in San Diego, as you're pointing out, and they're doing some really exciting stuff in this sort of host pathogen evolution realm as well. He came from he was more of a biochemist as a grad student at UCSF, working on some HIV stuff, and then added the evolution angle.
So Lost Laboratory has been, Jody is her first name. She's been listening to TWiEVO for 8 years. She said one day, Matt said to her, he was talking about chikungunya, and she was freaking out. She thought it was so cool. He said, there's a podcast you might like. And she said, I started listening, and I haven't stopped, and now she's doing science because of us. So I think she used to be a graphic artist, so I think that's a great story.
Really cool.
Now, Matt, to close this circle, I had a student in my virology class a couple of years ago who was very good. He used to sit in the front row, and he ended up going to Matt's lab to do his PhD.
Cool. I love it.
He just sent me, this past August, he defended. So this is all a very small, intertwined world, right? And I'm very proud to be part of it, You and
me both. Yeah. But we got to get to Larry's listener pick. He's got a recommendation.
A year-old episode of PBS Eons, The Genes We Lost Along the Way. I think it does a really good job explaining genomic evolution at an introductory level with clear examples like GULOP, UOX, and genes encoding for sensing bitterness. Probably most of your listeners already grasp what's covered, but anyone who's uncertain or friends and family will likely enjoy this, and he gives a link for that. Love the show. Thanks so much, Larry. Thanks, Larry.
Oh, yeah. Wow. I hadn't heard about that. I love that title, The Genes We Lost Along the Way. And yeah, that's, we kind of think about in evolution, what are we making? But speaking of Seattle, Maynard Olson, one of the now long retired geneticists, had this idea of less is more evolution, that they're like big leaps from losing things. And, and we kind of talked about that in the balancing selection or the trade-off. If you might let go of an immune defense and enjoy some benefits, but then, the gene that has some impact, not what you gain, but also what you lose. Yeah. Very cool. Thank you, Larry. I'll take a look at that.
All right. A couple of last comments here from folks. Pete says, doesn't Rich Condit owe us a final talk? What do you mean a final talk? Where's he going?
Rich will be talking about pox viruses all the way through for years to come. I'm anticipating, yes. Lots of good stuff.
The Eddie Holmes TWiEVO was 940. Thanks, Les.
Thank you. Yep.
And here's the paper I was thinking of. Thank you. Parallel adaptation of rabbit population to myxoma. Yeah.
Exactly.
Thank
That was
you.
the one I was
Thank you
thinking
very
of too.
much.
Yep.
Um,
Thanks, Terry.
all right. I think that does it, Nels. We, I mean,
We
I
did
have
it.
trouble leaving because I enjoy talking, but we have to get back to other things. So that's
Life
been
goes on.
TWiEVO 84. Oh, by the way, someone said here that link to show notes is not working.
Oh,
Yeah. So I haven't set up the show notes yet, and I won't until I get the audio and edit it, which I
yep.
may do right now. But yeah, I shouldn't have put that link in there because it's not live yet. So just it won't even be live once I end this stream. It has to, I have to go in and make a post. So
handcrafted podcasting over here.
yes, this is handcrafted. Yeah, what is it called? It's artisanal podcasts.
Bespoke.
Because I'm doing it bespoke because I'm doing everything, folks. I don't want to do it forever, but one day we will. I have to do this. Anyway, that's TWiEVO 84. The show notes are going to be at microbe.tv/TWiEVO. You can send your questions and comments to TWiEVO@microbe.tv, but mostly you should just come here to the live stream and pipe
really
in because
fun.
we enjoy having you. And if you enjoy what we do, we'd love your financial support. That's how we do this, and that's how I do this going forward, and we need resources to do this. Microbe.tv/contribute. Nels LD is at selvolution.org. He is L Earlybird on Twitter. Thanks, Nels.
Yeah, thank you, Vincent. Great to be together. Thank you, everyone who showed up. Great comments and questions along the way. Walking with a bounce in my step as the afternoon goes on here. So looking forward to the next time.
I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiEVO is by Trampled by Turtles. You can find their work at trampledbyturtles.com. And speaking of music, there it is.
There we go.
You've been listening to This Week. We got to lower that. It's a bit too loud. Hang on. There we go. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious. Wow, look at that timing, Nels. How about
Beautiful. I love it.
that? Thanks, everybody. Thanks, mods.